This is an explanation of the bacteria lab that we will be doing. It's an original design lab, so there's basic steps, but you have to figure out how you want to actually go about it, and everybody will do something different. So when you're reading the steps, there's part 1 and part 2. So part 1 is looking at specific environments. So you're just going to plate from a single environment. And for part 1, what you'll end up doing is you'll compare the bacteria load, how many bacteria and what types and that kind of thing, wherever you decide to sample initially. And you'll compare that to 3 or 4 other members of a group that you'll pick or I'll put you in. And so that's how you're going to compare specific environments. So really it's part 2 that you're mostly coming up with your own experiment on. So part 1 describes how to do the plating, and I'll describe that after what you're supposed to do for part 2. So with designing the experiment, the whole idea is that you're going to take an environment. Now it doesn't have to literally be a surface or anything, but it's some environment or object or place, something like that, that you're interested in seeing. And you're going to have 3 different platings from that environment. So one plating is the original. So this is the original environment. And then you'll have 2 treatments. So this side will be treatment A, let's say, and this will be treatment B. Now there are no specified treatments, and by treatment you don't even have to like be adding anything. It's just you're going to take this original environment, wherever you got that from, and you're going to do 2 different things to that environment. Now you'll need to make it to where you can treat half of the environment one way, and then that same environment treat the other half another way. So you know, the classic thing, right, is some kind of cleaning supplies on some, 2 different types of ways of cleaning on a surface. There's lots more interesting things you can do too. You just have to think about it. So what's going to end up happening, and this is a poster you're going to do. It's like a quasi-formal lab, just not a different sort of write-up. So you're going to want to come up with something good because you share it with the class. Everyone comments on each other's. You get graded. Some of that's along the lines of creativity of the design of the experiment. So let's say you decide to plate. So I'm going to look at the bathroom door handle. It's kind of a classic but not super exciting example. But you go to the bathroom door handle and you have the door handle. And it's the best I can do. And you're going to, I guess I should put this key, that is a keyhole. Yeah. And what you'll do is you'll take your swab. So you have this cotton swab, right? It's a sterile swab. So when you take it out, you're not going to want to touch it to anything. You're going to hold the end of it over here, and just right before you swab is when you'll take it out of its sheath. And so what you'll do is you'll take that swab and you will swipe it across the surface that you're sampling. Swipe it, across that surface or object or place. And then what you'll do is, without moving it around in the air too much, because you'll pick up other bacteria, is you're going to take your plate. So I'll make our plates green. So here's our plate. This is the agar plate. We're looking down on it. And if you look at it from the side, there's a top to the plate and then a bottom to the plate. So the top fits over. This is the top. It's the lid. The lid does not have the agar on it. The agar sits on the bottom of the plate. And that's obviously where you're going to spread your bacteria. It's important to recognize which side of the plate the agar is on as well, because that's the side you want to label on. So you don't label on the top, because if you label on the top can come off. It does. It comes off. And so you can mix up tops. And I actually have a video linked to online that shows that. So what you want to do is you want to label on the bottom of the plate. So the side that the agar is on. And the way you'll label is around the edge. So you'll put your initials, and then you'll put your environment. So this is the original bathroom handle. You know, you'll know what it is, but you want to make sure you know which treatment it is or whether it's the original. And then we should put the date. You notice how you can actually write the date, how I'm labeling all around the edge there. You don't want to label in the middle, because if you do that, when you're trying to look through the plate to look at the colonies that are growing on there, if those are bacterial colonies, you're not going to be able to see anything because you've written all over the bottom. And like I said, you also do not want to label on the lid. You want to label on the bottom of the side where the agar is, because that lid comes off and then you won't know, you can mix them up. And so if you label on the bottom, that doesn't happen. All right, so you swipe your surface, and then you're going to take the lid off, and you actually, if you can, don't even remove it completely. You'll just kind of crack the lid open, and then you'll take your swab, and then along the surface, we're just going to do a very simple plating. There are more sophisticated ways to do this to spread out the bacteria. You take microbiology, you'll learn those. But what we're going to do is just going to take your swab, and you're just going to spread it across the surface. You don't want it to just be like that. It's not going to spread it enough, but that should be sufficient. And so then you put the lid back on. So after you've cracked the lid open, you spread across the bottom. You don't want to gouge into it because it's, you know, like a little bit harder than Jell-O, but it will crack and it will get gouges, and so that will make your bacteria grow all crazy and not, I mean, they'll grow in the cracks and it will be all difficult to tell what's going on. So you just want to gently spread it across the top, and then you're going to close the lid again and you'll throw your swab away in the trash. So then that's the original plating, and that can go on here. So then your two treatments. So whatever, let's say I decide to take my doorknob and I decide I'm going to do two things to it. I want to see what the effects of, you know, cleaning it with water and cleaning it with salt water. Okay, so you'll take half of your doorknob and you'll clean that with, let's say this is the salt, the water, regular water. And then you'll take a new swab, and then you'll take that swab, this is a new one, and you'll swab that half and plate it in the same streak fashion just on half the plate. And so that you know where the split happens actually prior to plating on that plate, you should split it. So what you'll do is, here's your plate that's going to be the treatment plate. So you'll actually literally take a Sharpie or permanent marker, and on the bottom, the side with the agar, on the bottom side, you're going to draw a line approximately across the middle. And that line is going to be where you do not cross when you're swabbing. So after treatment A, and after you swipe this treated area, and you're going to just plate that treated on that side. And of course, before you did that, you'd want to do your initials, and then you'd say treatment A, and I'd just put water on there, and then you put the date again. And then on this side, you want to put whatever your treatment B is, so I'd put salt water. And then you'll treat that second half of your environment with whatever your second part, your second treatment is. You notice I didn't treat the same environment twice. You'll take your third swab, take that out, sample the environment, and then you'll take that and plate it, not crossing the line on the other half. I guess I should go this way. On the other half of the plate. And this way you'll have 3 environments, and we can save a plate. So take your third plate if you haven't used it already, and bring that back to me at school so that we can use less plates, because this is just as efficient. A lot of times, actually, we'll split plates up into 4s, quadrants, and plate different things on each quadrant. I've even seen ones that are like 6s, and you just plate like maybe one colony or one smear in each of those. So it's not unusual to do this. It's actually done in lab all the time. So That's the basic idea. So you have your original environment, then you treat one part of the environment one way, one part the other way. Like in this case. And please don't use this example. This is not even a very interesting one. But then you'd see the differences between those two when you observe later. So I've also seen, instead of like a treatment to try to get rid of bacteria, people do different kinds of treatments to see what kind of more bacteria will grow. So you can think of it the opposite way, maybe not trying to get rid of it, but trying to see if you get more of it, or different types of bacteria depending on how you treat the environment. And there's all sorts of stuff outside that could be interesting. And so you're just going to, in the end, want to be able to make comparisons between what did it start as, and what did the bacteria look like. Then after you treated it one way, how did that change it? After you did another way, a different way, how did that change it? So you'll compare those two sides to each other and both sides to the original. So then once you're done with that, you're going to take all your plates and you'll actually put them upside down. So that way if any condensation forms, you won't have it dripping onto the colony. So the condensation always forms on the lid. So if you put your lid face down, that means your agar is up, so it's not going to get any drips on it if the lid gets condensation on it. So you're actually going to put them upside down, and you'll stack those three like we did when you took them home, and then there'll be the third one. Then you can tape them up again. When we get back to lab, I'll show you how to use some parafilm. If you got parafilm when you went home, it's just a little piece here. It's sort of like wax, but a film wax. And so you just are stretching it gently. It'll stretch pretty neat, actually. And you put it on one edge of your crack between your lid and your plate, and then you'd stretch it around the whole plate until it comes back to the beginning. If you don't have parafilm, we'll do that in class, just to seal it so it doesn't dry out and so you don't have any bacteria escaping. I'll put up a video about them using parafilm as well, and you can see that. All your swabs need to go in the trash can, and like I said, when you're plating, you don't want to open the plate all the way if you can help it, because that exposes it to bacteria in the air. And you also want to be careful not to crack them because they are just plastic. And if you crack it, then bacteria from the environment will get in there and it'll mess up your experiment. Also, if you drop the plates, the agar can crack, just like if you were to drop, Jell-O. It's like I said, it's a stiffer Jell-O, but that can break. And so if that happens, then you're going to have problems as well. So you just need to be careful with your plates, and they'll get, they can get crushed in a bag, so you'll want to pay attention to those. But for bringing it home, if you don't have the parafilm, you'll just tape around it like you did bringing it to your house or to whatever environment you're going to. Okay, so that's the basic idea. I hope that helps you understand where we're going with it. And like I said, I'll post a couple other videos with some techniques as well. But this is the basic technique that we're doing with just the streaking straight across the plate.